Alright guys, if you have two sets of cell divisions or you have two sets of cells undergoing divisions, and if you see the different divisions are going in different directions, you might wonder why they are like this. You might wonder why this one is going like this, then like this, then like this, and this one is just straightforward, and finally we have a division like this. Okay, yes, it is very simple. They are very simple. Now this is what we call mitosis in biology or molecular biology or cell biology, and this is what we call meiosis. This is what we call meiosis in cell biology. Now, what is the difference? They look similar and students have often confused them over the years. Now, my name is The Analogous and I am here to break it down for you. Before we continue, make sure you like this video, you subscribe because it will be helpful to you, and make sure you stay till the end of this video. Alright, let's dive into it. Now, quickly, what is mitosis and meiosis? Quick difference. Now, these things are cell divisions. Now, cell divisions are processes in which the body grow. Now, when you were smaller, they've not added any cell to you. No part of you has been added to you. Nothing has been added to you. Just what happens is that you have some set of cells in your body when you were smaller, and those cells keep dividing and dividing, and they become more and they multiply, and it gives rise to the big you now, or the adult you, or the teenage you now. Now, these divisions, or the way these cells multiply or divide into many forms, are in different ways. Now, these two ways are majorly mitosis and meiosis. Now, what's the huge difference? Mitosis occurs in all body cells called somatic cells. Cells that do not undergo, that are not reproductive. Now, while meiosis do not occur in all body cells, it occurs solely to reproductive cells. That means cells that can undergo mitosis. Cells that can that are reproductive, that have the ability to join together and form a new organism. So, sex cells, and this meiosis results in the formation of what we call gametes, such as sperm and eggs. Now, mitosis results in the formation of other body cells, which we call somatic cells. Now, another big difference is that at the end of meiosis, in most cases, though some few exceptions, we have 4 daughter cells. At the end of mitosis, in most cases, we have 2 daughter cells. So, I'm going to show you the difference. Now, another thing is that mitosis just results or give rise to diploid number of chromosomes. So in mitosis, diploid number of chromosomes is formed. In meiosis, haploid number of chromosomes is formed. Haploid numbers of chromosomes formed. That means there's reduction in your chromosome number. That's why when a man and a woman combine, when they meet, the male is going to produce half number of chromosomes in his gamete, and the female produce half number of chromosomes in his gamete, and you produce diploid number of chromosomes, which will now result to you or any other person. So that's the major difference between meiosis and mitosis. Now, quickly, let's dive into it. Let's dive into mitosis. Now, first, they are called cell division. Something occurs before cell division. Now, in the cell cycle, 2 things occur. Yes, in the life cycle of every cell, 2 things occur. Now, the first one is what I call interphase. Now, or what many people call the resting stage. It's into 3 steps. There's G1, there's S, there's G2. Okay, in an advanced video I will explain. Now, for the other part of the cell cycle is cell division. Now, cell division now includes mitosis and meiosis. Okay, now in the cell cycle, the cell is basically not dividing, not necessarily resting, not dividing. What's occurring there is the replication of DNAs, the transcription of DNAs to produce RNAs, therefore translocate to produce proteins, and so on and so forth. That's what we call in interphase. Okay, that's what now in cell division. Now the cell has gotten to a stage that it now want to replicate its kind. Do you get? So the first one of the divisions that occur is mitosis. Now, mitosis, I already told the difference. I already told you what happens in mitosis. Diploid number of chromosomes is formed, and mitosis is into these stages. Please take note. It's called PMAT, so that you don't forget. So mitosis is into these stages. PMAT. P stands for prophase, M stands for metaphase, A stands for anaphase, T stands for telophase. So what happens now? Mitosis results in the production of 2 daughter cells. So one cell of my hand is not a reproductive cell. Reproductive cells are found in the testes and the ovary for women. So now one cell of my hand, we produce 2 cells. So 1 produce 2, 2 produce 4, like that. So that's how it works. But the stages is PMAT. Now, Prophase. What happens in prophase? Prophase is the longest stage of mitosis. It is the longest stage of mitosis. Yeah, it can be divided usually into early prophase and late prophase. Early prophase, late prophase. What happens in early prophase? In early prophase, the things like the nucleolus. If you are used to a cell, you can check the videos on cell. Now, the nucleolus in the nucleus of the cell is going to disintegrate. The nucleolus disintegrates in early prophase, and the chromosome becomes more visible. Before, in a cell in interphase, this thing called chromosome carry our DNA. This thing called chromosome are usually invisible chromatin thread. They were invisible chromatin thread. So, but as you proceed to, as we get to prophase, early prophase, the chromosome begins to shorten and condense. They are not visible. Now they are not visible for early prophase. In late prophase, they start to pair up. So you get the chromosomes start to pair up to form, not necessarily pair up, they become more condensed and they form chromosomes. So this chromatid, remember they are invisible chromatid in interphase and they become visible in cell division, which is prophase, and they form what they call chromosomes. So this chromosome becomes more and more visible in late prophase. And now the nuclear membrane, this nuclear membrane of the nucleus is going to disintegrate in late prophase. Now, in late prophase, in early prophase, the centrosome are organelles that help to produce spindles, which help to divide the cell. This thing you are seeing is called spindle. So, in early prophase, the centrosome, this is a pair of centrole, it moved to the equator. And when they move to the equator, they start to produce spindle fibers. Do you get? Start to produce spindle fibers. All these occur in prophase. Now, in what's it called? Before on getting to metaphase, the nuclear membrane has disintegrated as a late prophase. Nuclear membrane is disintegrated. Now, getting to metaphase, metaphase into early stages and late stages also. But I'll just do a summary video because I'm working with O level now. So when I get to do my advanced, when I have to do my mitosis as a video, I get to do my meiosis as a video, I will expatiate better. So, but on metaphase, which could be early or late. Now, the spindles now have successfully the centrosome has successfully produced spindle fiber on early metaphase. They produce spindle fibers successfully, and these spindle fibers get attached to the chromosome. They get to the chromosome. They attach to the chromosome, particularly the centromere of the chromosome. So they attach to the chromosome, and they successfully in late metaphase help to arrange the chromosome at the equator. So the chromosome is successfully arranged at the equator. Now, this is important so that the genetic information that will be divided into the two cells, because we are going to have a cell, we are going to have a cell. Genetic information that will be divided into the two cells will be equal, so there will not be partiality. So that there's this arrangement of the chromosome at the equator. So this occurs in metaphase. Now moving to anaphase. At anaphase, the chromosomes at the centromere begin to split up at early anaphase. They begin to split up at early anaphase. At late anaphase, the chromosome successfully gets to the poles. So this homologous chromosome, they start to split up into chromatid. They start to split up into what we call sister chromatid. Which you should take note, 2 sister chromatid make up a chromosome. That should be noted. So they successfully start to split up and arrive at the poles successfully at late anaphase. So in anaphase, the chromosome split up at the centromere with the help of the spindle. So these spindles pull them. So the centrosome allow the spindles, use the spindle to pull them. The centrosome allow the spindle to pull them, so they arrive at the poles, and that occurs in anaphase. Remember, the goal is to divide the cell and have equal genetic material. Now, on getting to the poles, they successfully get to the poles at late anaphase. On getting to the poles at anaphase, we enter a phase we call telophase, which is the last stage. Now, in telophase of prophase, in telophase of mitosis. In terms of mitosis, it's also into early telophase and late telophase. In early telophase, the chromosome or the chromatid, arrive at the poles. They arrive at the poles. So in this first cell, this upper cell, this chromosome, how would I put it? Okay, let me just do something here. So let's say these are two cells undergoing the cell division. So it has not yet been divided. Okay, the chromosome, the chromatid, arrives at the pole. They arrive at the pole. When they arrive at the pole, and they begin to start. They begin to condense. Don't remember they condense in prophase. They begin to condense or uncoil. still in early telophase, and they begin to uncondense, uncoil, and before you know, this chromatid begins to develop what they call a nuclear membrane again, and in late telophase, the nuclear membrane is successfully developed, and you start to see the nucleolus back. So this nucleolus that disappeared begins to fall back and begins to appear back, in late telophase, you see that the spindle fibers here, these spindle fibers now in early telophase begin to disintegrate, they begin to disappear, and they successfully disappear in late telophase. Okay, these spindle fibers disintegrate. Now, this thing you call centrosome, that produce spindle fibers, this thing called centrosome, they duplicate themselves. So in late telophase, they duplicate, centrosome or central duplicate themselves, so that you have pair. Because if you want to do cell division, okay, you obviously have a pair upward and a pair at the various points. So they duplicate themselves in each cell. So I'm not supposed to circle it, but just taking the centrosome, a pair of central is what you call a centrosome. So a pair of central, they are at right angle to each other, very close to the nucleus in a cell, not in cell division. Okay, the central, which in pair gives the centrosome, duplicate themselves, so that it will be needed for other cell divisions in time to come. And before you know, the cell, what's it called now? The cell begins to split up, and this splitting of the cell is what you call cytokinesis. So this is more like, I should call this telophase stroke cytokinesis, where the cell begin to split up, cytokinesis. So telophase, so cytokinesis. So that is for mitosis. Very simple. Now quickly, do not waste your time, let's talk about meiosis. Now for meiosis, quickly, meiosis is usually more complex than mitosis. Okay, usually more complex than mitosis. So I'll take my time to explain. Now meiosis has two forms, two stages. Now the first stage is what you call the reduction stage, and the second stage what you call the division stage. The division stage is very similar to that of mitosis. So the division stage of meiosis 2 is similar to mitosis. Please don't forget that. And I will tell you some things about it. Now, first stage is the reduction of chromosomes. Remember, in meiosis, the chromosome number is reduced. So we have, instead of having diplet chromosomes, we have haplet. For instance, in man, we have 46 chromosomes. Okay, after meiosis, they said we have 23 chromosomes. So there's a reduction in the number of chromosomes to call meiosis 1. Okay, now what happens in meiosis? Now, meiosis is also similar to mitosis. The same acronym, PMAT, are the processes that occur there. Prophase, metaphase, anaphase, and telophase. Just that meiosis will be 2. We have prophase 1, metaphase 1, anaphase 1, telophase 1. Also, we have prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, now for prophase 1, it is the longest stage of any form of cell division. Prophase is the longest stage of cell division, but prophase 1 for meiosis is the longest stage. Now, prophase 1 is into other stages such as Lixil-Padid. Okay, now there are other stages of prophase 1: Liptotin, Lixil-Padid. Liptotin, L is for liptotin, this one is for zygotin, this one is for pachitin, this is for diakine, this is for diplotin and diakinesis. Now remember, I said I'll do video on just meiosis. Then I will explain these things. These are the 5 steps in prophase 1. I will then explain this. So this is for advanced chemistry. Okay, this is for, I say advanced chemistry, advanced biology, or people in A level. For those of us in O level, you might not need this. Those in A level can also watch this video and learn. But everything is not thoroughly said. I'm just contrasting mitosis and meiosis. Now, quickly, for prophase 1, what happens is that very similar to the former type of prophase, but yeah, as usual, the nucleolus disappear. The nucleolus in any prophase, the nucleolus disappear. The spindles begin to the centrosome, okay, central in pair, they begin to migrate to the poles. Central begin to migrate to the pole and start to generate spindles in early prophase 1. Now, in late prophase, now still in early prophase. Now the chromosome begins to pair up. So homologous chromosome after condensing, they begin to pair up. About when we're about to enter the late prophase, so they begin to pair. That means chromosomes begin to stay with each other like in pair. Oh my god. So they begin to stay with each other like in pair. They begin to pair up. So this is pair. You see, they begin to pair. This pairing up is called synapsis. It's called synapsis. Now, one of the chromosomes that pair up, they are called these two chromosomes that pair up, they are homologous. These two homologous chromosomes, they pair up, and one from the father, one from the mother. Do you get? So one from the father, one from the, so this is called synapsis. So when they pair up, I think they pair up at I think zygote. Yeah, they pair up at zygote. That's synapsis or called zygote. And with time, this pair becomes shorten, become more and more visible at Pachitine, and diplotin late prophase is all called crossing over. In diplotin, it's all called crossing over. So the chromosomes they cross over like this. The point in which they cross over is called a chiasmata. They begin to cross over, and they cross over at a point you call a chiasmata. Now the crossing over is very important, so that now remember these chromosomes that are from the father and this one is from the mother. So these chromosomes that cross over here at the chiasmata, they lead to variation. So lead to variation and production of more favorable genes in the environment. Do you get? So this pairing up is called crossing over. It's very important, and crossing over leads to variation. Don't forget this. And finally, this pair that cross over, they start to separate again, or they begin to start to separate. This pair that cross over, they begin to start called diakinesis. So that happens in late prophase. And finally, the nuclear membrane disappears thoroughly. Nuclear membrane disappears. Now you can also see the chromosomes that pair up. They're also in pair here. So in metaphase 2, as a summary of metaphase 2 is that this pairing up of these chromosomes that pair up during crossing over, they are now held together. They are still held together, and they are now attached to a spindle, attached to a centroid via a spindle. So the spindle attach them at both ends. So one spindle to one, another spindle to another one via the centrosome, and another spindle. If you see, so they are attached to the spindle, they are arranged now at the equator. That occurs at metaphase 2. Now, in anaphase 2, in metaphase 1, that occurs in metaphase 1. I'm sorry, metaphase 1. So what we call them now bivalent chromosome. The bivalent chromosome, the ones that occur in pair, are arranged at the equator. Bivalent chromosomes, now not just homologous chromosome, bivalent homologous chromosome are now arranged at the equator. They are arranged thoroughly. When they are arranged at the equator like this in metaphase 1, the goal is to separate them and reduce their chromosome number. That was the goal for this stage. Now in anaphase 1, the chromosome begin to migrate away from themselves. They begin to migrate away from themselves, and this is done with the help of the centrosome. Centrosome use the spindles to put them, and each chromosome begins to go to each of the pole. They begin to go to each of the pole, and when they get to each of the pole, in telophase, they finally arrive at each of the pole, and they are separated. So telophase and cytokinesis will occur. So these two cells will more like they are going to separate like this. You get? They arrange at the pole, and what happens is that the spindles begins to disintegrate, they begin to disappear, the spindle begins to disappear, and that's it that occurs in telophase again is that the centrosome duplicate themselves. The centrosome begins also to duplicate themselves. Spindle fibers disappear, centrosome begin to disintegrate themselves. Now, in some cells, nuclear membrane begin to appear. In some cells, not all cells. In some cells, nuclear membrane will not appear, nucleus will not appear. It will just move directly to prophase 2. In some cells, nuclear membrane will appear, nucleus will form like it is undergoing its first mitosis. So nuclear membrane will appear, but just that what will happen is that this cell now that is formed, these two different cells that are formed, they have a reduced chromosome number. So the chromosome numbers here, the goal of this step 1 or this prophase 1 of this meiosis 1 was to just reduce the chromosome number. That's the goal. So the chromosome number has been reduced. Then now we move to prophase 2. Now, what happens in prophase 2? What happens in prophase 2? Prophase 2 is quite, pardon me, in meiosis 2 is very simple. So from here is meiosis 2. So for here is meiosis 2. What occurs in meiosis 2? In meiosis 2, for some cells that nuclear membranes and nucleus were not formed, they just proceed immediately into meiosis 2, and before you know, we're in metaphase 2. So in most cases, in some cells, prophase 2 could be skipped. In some cells, prophase 2 or telophase 1 could be skipped in some cells. Do you get? In some cells, telophase 1 could be skipped. In some cells, prophase 2. So it's hard to skip prophase 2, but prophase 2 is very important. So in most cases, telophase 1 could be skipped. That means there's no formation of this nuclear membrane and the nucleus again. What should just happen is cytokinesis and move to prophase 2. So telophase 1 could be skipped. Now, so for meiosis 2, it is called the division stage. Now, the cell want to divide into 2, it's going to further divide into 4. Now, but these 4 now will be haploid in number, they are haploid cells. So the division stage now occurs like this. For prophase, spindles begins to move to the poles. Then the spindles duplicate themselves and it begins to move to the pole. They move to the poles and they appear at the poles and they start to generate spindle fibers. And the nuclear membrane begins to disappear. Nuclear membrane disappears finally. That's the end of prophase 2. The nuclear membrane disappears. Nucleus already disappeared in early prophase. So nuclear membrane disappear, and it's ready to go. Now, before you know, it gets to metaphase 2. In metaphase 2, this early metaphase, the spindles get attached to the chromosomes. Remember, these chromosomes are haploid number of chromosomes now. So they attach to them and arrange them at the equator. It is necessary to arrange them at the equator so that the information between the cells is shared equally. So now it occurs in the first cell, it occurs in the second cell. It occurs in the first cell, it occurs in the second cell. So now this is where I drew it and stopped, and this is metaphase 2. So can I now talk about this metaphase 2? I can now talk about what's it called? PMAT A, anaphase 2. So in anaphase 2, what occurs in anaphase 2? So in anaphase 2 is similar to anaphase. Remember, it's very similar to this cell. So in anaphase 2, what occurs? So for each of the cells, remember there are 2 cells now, more like 2 cells. For each of the cells, their homologous chromosomes are going to, or their sister chromatids are going to divide away from their homologous chromosomes. So the sister chromatids start to move to the poles. So the sister chromatids, by the help of the centrosome pull them to the poles. Centrosome pull them to the pole. So the centrosome will pull the homologous. Okay, we have something like this, something like this. Okay, we pull them to the pole. So this is anaphase 2. So the sister chromatid begins to migrate now to the poles to go and form their individual cells. Remember, we have to establish something like this at the end of the day. They begin to migrate to their poles. These are the sister chromatids. They begin to migrate to now two sister chromatids make up a chromosome. They begin to migrate to their poles along their centrome. They split along their centrome. The centrome is like the point of attachment of two chromosomes. Yeah. So they begin to split they split along their centrome and they migrate to the poles. Now, finally at telophase 2. So finally at telophase 2, which is very telophase 2. So finally at telophase 2, what happened is that they have successfully gotten to the cells they've successfully gotten to their destination, and before you know, the spindles begin to disintegrate. For telophase 2, and the chromosome got into their destination, begin to form. So now we have 2 cells. So each of those cells, we have 2 cells. The chromosome. So these are the chromosomes now. They start to form nuclear membrane around. So 2 cells. So by that time, the chromosome must have gotten to their destination, and they start to form a cell membrane. A nuclear membrane around the chromosome, and before you know, you have a nucleus. And this occurs here. So that's what I'm trying to do. So you have a nucleus. So you can see it occurs in the first one, it occurs in the second one. So you can now see you now be having 4 cells. So you see, you're having 4 cells. So it's how we said meiosis results in 4 daughters. So meiosis results in 4 daughters. So the spindles disintegrate, there's the formation of the nucleus back, and cytokinesis occur finally, and that's what occurs in telophase 2. So I know this video is helpful. It's helpful to both O level and A level students, to be very sincere. A lot have been said in this video, and what you can do for us is to just subscribe to this video, and we'll see you in our next video. God bless you.